Hi, welcome to Detours Understanding Acquired Brain Injury. This is Unit 4, Chapter 3. So the question is, why do genetic testing in the first place? This seems like a silly question, but it's a valid one. So what are the reasons for testing? Obviously, family history of genetic disorders. If your family has a history of suspected or known genetic or chromosomal disorders, that would be a reason to perform genetic or chromosomal testing, predictive testing, to determine the likelihood of developing a genetic or chromosomal disorder. There are a lot of disorders where we didn't know that they had a genetic or partially genetic basis. And as we've made a lot of discoveries, many chronic illnesses we're finding do have a basis in genetics, at least partially so. Certainly the environment may contribute, but we're finding out that things that we once thought were strictly due to environmental causes have a genetic basis. And so this would be a strong reason to engage in predictive testing. Prenatal testing. Now, we're all familiar with this one. This is to determine the likelihood of the presence of genetic or chromosomal disorder in a developing fetus or during pregnancy. Newborn screening. This one is to determine the likelihood of a baby developing a genetic or chromosomal disorder during their development or at some point later in life. And this is something to understand. Not all disorders that somebody inherits are going to necessarily present early in life. It still may be chromosomal or genetic disorder, but it may present its symptoms much later in life. A lot of people think developmental disorder equals something in childhood. Not always. And you may want to look for symptoms of certain disorders that develop later. Also, newborn screening as well. And these are common. And we've been newborn screening for a long time. But if we also do genetic testing, we may be able to catch disorders where a dietary modification or some other kind of treatment, if we know ahead of time, we may be able to have mom modify her diet and to help prevent or reduce the likelihood of illness in the newborn, so it's less likely to cause problems even further down the line. Pharmacogenetic testing. This relates to something we had mentioned before, and that is to find out how certain medications will work or if they won't work ahead of time. And so if somebody does have a disease or disorder, find out which medications work. Pre-implantation testing, identifying genetic conditions in embryos before they're implanted in situations like IVF, things like that. Pre and post genetic treatment testing. With more and more gene-based treatments, we'll do testing to see whether the genetic treatment is taking hold. As we lack cures for many diseases and disorders, genetic testing is fraught with ethical, moral quandaries. And so we have to ask questions about, what do we do with this information? Some countries pressure parents then to abort, especially systems that rely on socialized medicine. You may say, well, gee, wouldn't this be more of a problem for here in the States? But there are many countries in Europe that have made claims like, gee, we've cured this disorder. And what they actually mean is they have pressured parents to not have children if they manifest a certain disorder. I'm not going to dive too deeply into this one. This is something we can discuss in ethics. This is a flashing light right here for ethical issues. But we have to decide as a society, as humanity, what life is worth for somebody with disabilities or with genetic disorders, because that's a slippery slope. And we've, as humanity, have traveled down slippery slopes like this before. So just something to keep in mind as we talk about genetics, what the implications are. Just something to roll through your brain as we talk about this. because we're still in a position where we have a lot of learning to do, and we're not able to cure a lot of things. That may change in the next coming decades. And that also presents questions of worth and what should be fixed, what not. What does a disorder mean? What is normal? What is not? These are hard questions. And the more you know about genetics, the more you're in a position to think about it, to perhaps provide answers through your choices for these kind of things. All right, enough of that for now, and we will move on to specific testing used by clinical geneticists and genetic counselors. There's a difference between chromosomal testing, as you're aware of what a chromosome is, versus genetic testing. Now, chromosomal testing includes something like karyotyping, which we've done for a long time. This is really the first type of testing available. Karyotyping is performed using a microscope, a photomicroscope, and they would take a photograph of a cell, and the chromosomal pairs would be matched based on their physical appearance. And then they would line them up into groups. And, this is the first pair, second, third, fourth, fifth, all the way through the sex chromosomes as a 23rd pair. And they identified chromosomal abnormalities in appearance, like extra chromosomes or a missing chromosome, or maybe a fragment is broken off, or things like this. And this process is called karyotyping. This has been around for almost a century. And so it's, you could derive basic information from this test. Things like identify Down syndrome, Turner syndrome, Pateau syndrome, and things like that. Another kind of test is the fish test. And I'm not talking about how well you catch fish here. I'm talking about fluorescence in situ hybridization test. This uses a fluorescent marker probe, binds to specific DNA sequences associated with chromosomal disorders, and they show you abnormalities of chromosomal structures, such as translocations, where segments of DNA detached and reattached, and they form for translocations or rings, which we've discussed before. Chromosomal painting, used mainly in oncology genetics to highlight complex DNA sequences that have swapped in place by using specific markers that are fluoresced by painting it, and they have an affinity for each specific chromosome. Now, this requires advanced protein engineering to create one of these, and a fairly sophisticated lab with skilled lab technology and a talented lab tech. Although robotics is making chromosomal painting easier, it still requires skilled lab techs to operate the equipment. So this costs more to perform this. Now, there's also the array comparative genomic hybridization, and this relies on a microarray computer chip with several hundred kilobase probes. These probes are synthetically produced, and they will identify when they make contact with certain kinds of genetic markers, and this will cause them to light up. This is useful because it lets the geneticist know there may be chromosomal duplications or deletions, and which specific chromosome or chromosomes are adversely affected. Again, this requires a fairly sophisticated piece of technology to make. However, they can sell these, and then if you have the right reading equipment, and pick it up, but the actual manufacture of the equipment isn't as expensive, or the equipment to produce these is expensive, but to read it and to apply it isn't as expensive. So if you're a doctor who has the microarray chip, these are fairly cheap, but if you're the company that makes these are much more expensive. So then, looking at genes themselves, there are specific tests for individual genes, base pairs, for SNPs, the single nucleotide polymorphisms. And this list is in no way comprehensive, but ways to look at individual genes or SNPs. There's DNA sequencing. Now, this has become an automated process. This still can be expensive. There are different techniques, And if you want to look at this, this would become a whole video all by itself. A sequencer it'll read the exome pairs, all the base pairs that code for specific proteins and their neighboring pairs. Sequencer may miss genes that contribute to disorders. So, as not all disorders are caused by specific exons, which are responsible. Some are caused by various introns, which may affect how other genes are switched on or switched off. So, to keep in mind. DNA methylation analysis, which is similar to DNA sequencing, except DNA methylation analysis machine looks for DNA or RNA marked by those methyl groups, which turn on and turn off genes, by using probes or fluorescent or other detectable media that the machine looks for. So it adheres to specific methylation groups. And so they're looking for which genes are potentially switched on or switched off. And this helps determine epigenetic effects on the individual. Southern blot. This is an old technique which relies on a process called electrophoresis, where the DNA was physically separated based on its size, and it was tagged using radioisotopes. It still is used, but it requires, again, a very skilled lab tech, somebody who knows how to handle, and that increases costs because of labor and to train somebody to use the southern blot technique. However, it is effective if you know it's a long, heavy chain and you know the mass of how long that should be. And with a high degree of precision, you can say, oh, well, this is a variant allele. It's a little tiny bit heavier, more massive. It's marked by specific nucleotides with duplications, gene duplications, or deletions. It's lighter than it should be. Oh, this may be, missing this sequence right here. We know that's a variant allele. It's more likely to make this defective protein that we've seen before. So you're looking for specific gene sequence errors. PCR. This is, you should recognize this if you know a little bit about, like, when we were looking for COVID. PCR chain reaction tests. These are really cheap pre-manufactured tests used by doctors' office geneticists. And they do like the one-off screenings for single nucleotide polymorphisms, changes to DNA, RNA, markers on diseases. The antigens on their surface fit with a manufactured antigen that locks in and it kind of lights up. It can light up a strip or whatever else. It can cause it to turn purple, and these dime a dozen, these PCR tests. And they're pretty effective, but their specificity has to be fairly high. And so they'll detect like one thing. And that's why they're used. Many home tests involve these. The COVID, the RSV, pregnancy tests, looking for specific markers which become attached to the genes. And then they use a chemical amplifier, which bonds gelatin with a gelatinous substrate. And it may also be used if you have multiples of these, multiple PCRs lined up. You can look for like a couple hundred genes if you know exactly what you're looking for, and then they can either cause an electrical signal to flow if you have a computer looking at hundreds of these at once, or a fluorescent light to come on. Again, have a computer look at many samples, many different antigens, and you can have it scan for multiple at once. The chip itself would detect a fluorescence or an electrical current, which it passes through, and I see that this marker has been changed and it's more conductive, so that must mean yes, we have this antigen attached. So yeah, some automated microarray tests on a chip can be performed for really rapid screens or emergency or critical care. When you see these little like cartridges that pop into like a blood analyzer, that's really basically what it is. Microarray testing or PCR testing has really changed the testing industry. But you need a high degree of specificity for this to work. Then to look for genetic disorders, there's something called metabolic testing, which looks for the byproducts of a chromosomal or genetic disorder, looking for like these aberrant proteins, either when they're broken down or their products look different chemically. And so we look for unusual byproducts, altered sugars, or some kind of residue that is left over from when enzymes break down altered protein products. For example, amino acid testing. It's exactly what you think. Looking for elevated or suppressed amino acids, perhaps PKU testing, organic acid testing, cerebrospinal fluid, blood, sweat, absence or abnormal organic acids, enzyme testing, looking for unusually formed enzymes through X-ray crystallography, which is a method of using high-power X-rays to see how a protein or amino acid is structured. Does it have an unusual, what's called handedness, left versus right-handedness? How does the crystal put together? This is another way of telling whether there's a genetic disorder. Urine organic testing. Tests for abnormalities, for deficiencies, excess production, like Gaucher's, PSK, pyruvate, and lactic acid testing. Again, elevated levels are suppressed, telling you that there's something genetically wrong. Many of these tests are for very specific conditions, or used to identify, especially the metabolic testing in infants, to search for specific disorders. So early treatment or planning can be used, or referral to a clinical geneticist, for example, for perhaps treatment, or a pediatrician or pediatric geneticist, or something along those lines, to improve testing and caretaking. There are other things you could do too. Medical pedigree. This is essentially an advanced form of a family tree, but it uses the principles of Mendelian genetics and plots through living and deceased family members and looking at what happened, what illnesses. This requires a lot of knowledge in how to properly analyze, past histories and information from that. And a medical pedigree requires skill and understanding how to organize it. The specialist assists a clinical geneticist or a certified genetic counselor. And this person is typically a medical genealogist who's trained in how to do this. Many medical geneticists or medical genealogists, are experts in genealogy, but they also have medical training, and they're certified in doing this. But a certified genetic counselor also has training in how to do this, and so this helps them plot out. To create a basic medical family tree or medical pedigree requires some knowledge of at least 3 generations and knowledge of the medical diseases and disorders which a person's family had, and then understand how inheritance of these diseases unfolds. This is probably one of the most helpful tools available to a medical professional, but this takes time and requires a lot of information, accurate information from the patient. So these are the tests, these are the tools that professionals use to help understand genetic disorders and figure out potential treatments. Eventually, we'll get into how to make a medical pedigree in more detail, and how to perform Punnett square, which is another tool of multiple trait size in the neurogenetics series to help you track down neurogenetic disorders potentially. And keep in mind, these can be helpful when you're working on your family tree. All right, so we will wrap up here for Unit 4. We'll see you in Unit 5. If you found this useful, please click like, subscribe. Don't forget the bell icon. And if you have any questions, comments, please post below. Share with anyone you think may benefit from this. We'll see you next time. Bye.